Today I will give a talk about this global analysis of glycoprotein chaperone that reveals that there is a intracellular lectins that regulate ER stress and unfolded protein response. So what is glycosylation? Glycosylation is a complex and ubiquitous post-translation modification, and around more than 50% of the proteins are and more than 50% of the post-translation modification is the glycosylation, and we can see the glycosylation that can be seen on the epithelial cells of the rat, and simultaneously we can see the brush-like layers on this glycolex layer on this bacterium. And this glycosylation also plays an important role in the biotechnology, as it helps the antibodies to get their native structure. As we can see the yellow color glycans that helps this antibody to get their native structure. And there are two major kind of glycosylation that are observed within the mammalian cells. First one is the N-Glycosylation that usually starts in the endoplasmic reticulum. Another one is the O-glycosylation that usually starts in the Golgi. And since our focus of this study is about N-glycosylation, we will understand how this N-glycosylation proceed within the endoplasmic reticulum. First, there is a dolicol precursor that is formed. Some part of this dolicol precursor, that is mannose and 2-glycanic part, will form outside the endoplasmic reticulum, and when after that it will move inside the endoplasmic reticulum, and this 3-glucose monosaccharide will added to this dolicol precursor. And when the proteins are translating within the endoplasmic reticulum, this dolicol precursor or this carbohydrate precursor will bind to the newly translating protein at a specific site, that is at aspargine site, with the help of this oligosaccharyl transferase. After that, the trimming of this glucose will occur via this glucosidase 1, and the trimming of another glucose will occur via this glucosidase 2. And at this condition, or carbohydrate at this conditions can be detected by the calnexin and calreticulum. These are the intercellular chaperones that are usually found in the endoplasmic reticulum, and that helps the proteins to get their native structure. At the same time, also helps the proteins to get their native disulfide bond. And if the proteins are not able to form their native disulfide bond, they will continue to circulate within this calnexin and calreticulum cycle to get their native structure. And if they get their native structure, they will move towards this Golgi apparatus for further post-translation modification. And if they are not able to get the native structure, they will move to this ERAD pathway, that is endoplasmic reticulum associated degradation. So what is the scope and significance of this study? So first question that we have asked is, what are the glycoproteins? How does this calnexin and calreticulum affect the glycoprotein biosynthesis? And the second question that we have asked, whether it will modify any cellular glycosylation pathway or not, and if there is any other function of this calnexin and calreticulum where it plays an important role. As we have found in the literature that there are calnexin and calreticulum related to several diseases, and also recently published study in the Science Advance reveals that this calreticulum knockout leads to the reduced packaging of this lentivirus. And also a recent study in the Cell Journal has pointed out that these common mutations among these myeloproliferative neoplasms has this homozygous calreticulum mutation, or we can say that this most of these myeloproliferative neoplasm patients have these calreticulum mutations that leads to the cancer type phenotypes within the patient. And thereafter, this calnexin also plays a important role, as it has reduced the it has reduced the spike packaging of the beta coronavirus that has been recently published, and also it is the prognostic, it is the marker for this colorectal cancers. So these are some of the things that are already been established or already been published in the already been available to the already been available in the literature, and we need to explore whether it has any further roles or not. This calnexin and calreticulum cycle has any further roles on these aspects or not. So how we proceed our study? We proceed our study with this HEC293 cells, and we prepared the bona fide knockout, that is calnexin knockout, calreticulum knockout, and also the dual knockout. And simultaneously we also prepared this MOX knockout, and then we did this we studied these cells on 4 different aspects. First we studied the proteomics analysis, and thereafter, since this calnexin and calreticulum are related to the initiation of the N-Glycosylation, so we hypothesized that it will also affect this glycomics profile. So we did this lectin binding and glycomics binding assay, and thereafter we also did this functional study based on this ER stress, as this calnexin and calreticulum knockout might dysregulate the functioning that is going on within the endoplasmic reticulum, that might trigger the ER stress. And finally we end this study by checking whether these knockouts have any role in the cell cycle or apoptosis or not. So first we did the proteomics analysis. In this, what we did is that we take the HEC cells, we lyse the cells and did the trypsin digestion, and then check them using the mass spectrometry, and then analyze the data using ion star. And these are the PCA plots of the top 50 variant, and this PCA plots indicates that all knockouts behave differently. At the same time, biological replicates are close to each other, so this also provide a indication that our samples are biologically reproducible. And thereafter what we have find out is that some of the proteins are dysregulated. We have detected around 4,800 proteins. Out of this, 0.7% in the calreticulum knockout are upregulated, and 0.5% are downregulated. And similarly, the proteins are upregulated and downregulated are been mentioned here. And we have find out that MOX has the significant number of upregulation and downregulation, and this could be because that MOX plays a critical role in trimming of the monoglucose monosaccharide, and that helps the newly translated proteins to get detected by the calnexin and calreticulum. And thereafter we did this pathway enrichment analysis with the help of this volcano plot, and the volcano plot is shown about this calreticulum knockout versus wild type. In this way, when we have analyzed the highly upregulated proteins, we can highly downregulated proteins, we can find that there is a downregulation of the general transcription factor. This is in line with the literature that that this general transcription factors will lead to the downregulation of the translation of the proteins that are responsible for normal translation process that are not essential during this abrupt condition. And we also see that there is down regulation of this glycosylated proteins, the O-glycan proteins. When we did the same thing for the upregulated protein, we have find out that there is upregulation of the chaperones, and this could be because of it tries to compensate the loss of or the absence of calreticulum in these cell lines. And simultaneously we also find out that there is a upregulation of these proteins, the ATP13A1, and this proteins plays a role in preventing the cross contamination of the mitochondrial proteins into the endoplasmic reticulum. And another important thing that we have find out that there are many UPR related genes that are upregulated So this could be the indication that there is a UPR response in this kind of knockout. And we did the similar analysis for all the different knockout, and these are the major findings that we have find out. There is upregulation of the unfolded protein response, and also some of the glycosylated related proteins are dysregulated. And finally, we have also seen that some of the genes that are upregulated that might cause apoptosis suppression in MOX and calnexin knockout. So therefore we decided to do further to check the unfolded protein response, or we can say the ER stress. So what happens in the ER stress, that these three signaling pathways get upregulated, and these signaling pathways will lead to the upregulation of the chaperones that helps in the protein folding, and simultaneously these other pathways, that is ATF6 and ATF4, also leads to the upregulation of the genes that are responsible for the ERAT pathway, that is endoplasmic reticulum associated degradation, that takes care of the unfolded protein response by diverting them towards this degradation via ubiquitation. And in this case, we have find out that we check this UPR response with the help of the fluorescence response that is already been developed, and we use this fluorescence response. We use this fluorescence to check the ER stress in the knockout, and this is the fluorescence reporter for ATF4 that we have used. In this case, when there is no ER stress, the translation gets terminated at ORF1 and ORF2. However, when there is a ER stress, a translation begins at ORF3 and terminates at after this NLS sequence, that is at the end of this fluorescence reported. And during this, it will also translate this M scarlet or the red signal. So whenever the cells are under ER stress, this M scarlet signal will get translated, and we will see the red signals. So when we did the fluorescence assay on different knockouts, we can see that different knockouts will have different kind of ER stress, as we can see the different fluorescence signals in all the knockout. And when we quantify this fluorescence image, and we can see that the calreticulum has the highest knockout compared to the other knockout. And we try to check these results using different instrument. So we check the results using we have also checked these results using the flow cytometer by measuring this red fluorescence, and we can see that we can see the same results, that is calreticulum has the higher ER stress compared to the other knockouts, and whereas this calnexin and MOX knockout has the almost equal ER stress. And thereafter, apart from this fluorescence reporter, we also checked a signature protein that is usually produced whenever the cells are under ER stress, that is CHOP protein, and the results are in line with our other two measurements, that is calreticulum has high ER stress compared to the other knockout. And thereafter, we did this lectin binding. Thereafter, we did this lectin binding study, where we take the cells and we use the two kinds of cells. First, we treat the cells with the neuramic or neuramidase silidase, and we use the cells without any treatment. And after that, we combine these cells using this fluorophore lectin fluorophore conjugate. We use 23 different lectin fluorophore conjugate, and after this 20 out of these 23 lectins, we are able to identify 4 different lectins in which we have seen the significantly upregulation of the signals, and all these 4 changes are found in the calreticulum knockout. And when we did the composition analysis of the different knockouts, we do not able to find any significant changes that are observed within the knockout. So this finally leads to a conclusion that there are not much significant changes in the composition. However, the structural changes have occurred, and this could be in line with the literature that calnexin calreticulum plays a role in providing the actual structure of the proteins. Finally, we did our complete our studies with this cell cycle analysis, and in this cell cycle analysis we did first we did check the apoptosis assay, and we are not able to find any significant changes in the apoptotic cells. Thereafter we also did the cell cycle assay, and there we find that some of the significant changes that occur in all the knockout, that is in G1 phase there is a down regulation of the G1 phase, and whereas there is upregulation has all been observed in the S phase for the calreticulum and MOX knockout. So this in overall provide a idea that this calnexin and calreticulum plays a role in regulating the cell cycle in regulating the proteins that are responsible for cell cycle maintenance. So what are the conclusions that we got? First conclusions is that calnexin and calreticulum plays an important role in maintaining the homeostasis within the of glycoproteins. That is, if any of one of this is get dysregulated, this might lead to the more number of unfolded proteins within the endoplasmic reticulum, and that might cause the ER stress. And since calreticulum being the ER lumen protein, it is possible for the calreticulum to be present at more places within the endoplasmic reticulum compared to the calnexin, and this could be the reason why calreticulum has more broad specificity, and this could be the reason calreticulum has more broad specificity, and disruption in its function can lead to ER stress. And finally, this we are able to get a final conclusion is that we are able to get the hierarchy of the ER stress and the quality control within the endoplasmic reticulum. That is, calreticulum has the higher ER stress than the MOX knockout, then the calnexin knockout, and after that the dual knockout. And this study, along with that, since now there is a push in the biotechnology products to move towards the cell lines that are human derived. So this study will help in those biotechnology products where post-translation modification is important, as the functioning of this calnexin and calreticulum might help us to design the cell lines that can produce biotechnology product in a more customized manner. So I hope you have understood what this study is about, and if you have any doubts, feel free to ask any question. That's all for this video. Thank you for your time.